Good afternoon. I'm Dr. Brian Shapiro, a scientific content specialist at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled Viral Metagenomics and the Use of Standards, From Biology to Clinical Applications, presented by Dr. Tasha Santiago-Rodriquez. Dr. Santiago-Rodriquez is a bioinformatician at Diversigen, Inc. In this presentation, Dr. Santiago-Rodriquez will describe how implementation of viral standards and controls can help determine biases that can be introduced at different stages of a viral metagenomics pipeline. Dr. Santiago-Rodriquez will then show how these standards may evolve to mimic simple and more complex viral communities in diverse environments. If you have any questions for our speaker, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. First, a quick word about ATCC. ATCC is a private, nonprofit organization founded in 1925. We are headquartered in Manassas, Virginia, but also have an R&D facility in Gaithersburg, Maryland. ATCC is a leading global supplier of high-quality biological materials, providing the scientific community with a diverse range of industry-standard products, including microbiome research solutions. Which leads us to the focus of today's talk, metagenomic standards for the virome. So with that, I'd like to welcome Dr. Santiago-Rodriquez.
Thank you, Brian, for the invitation. It is really a pleasure for me to be here and talk to you about viral metagenomics and the use of standards from biology to clinical applications. For today's webinar, I'm going to be talking about Diversigen, what do we know so far about virome research, biases in virome research, applications of mock communities in virome research, and the potential applications of standards in the detection of ongoing and future pathogenic viruses, considerations from SARS-CoV-2. Let's first talk about Diversigen. Diversigen was a company of Baylor College of Medicine and the Center for Metagenomics and Microbiome Research here in Houston, Texas, and it was founded in 2014. The team currently has a group of very talented people, including over 15 PhD scientists. As some of you may know, Diversigen is currently part of OraSure Technologies, which also includes locations in Minneapolis and Houston. Let's now talk about what we know so far about viromes before talking about biases and other potential applications of reference materials. As many of you know, the number of microbiome studies have expanded during the last 10 years or so, and thousands of studies have been published so far. But a microbiome area that remains largely unknown is virome research. As you can see here in this expanded graph, the number of virome studies has increased, but very, very slightly. These numbers are showing that virome research is still behind microbiome research. And there are a number of reasons why this is, and include, for example, the difficulty of studying viruses, probably because of the very small sizes compared to bacteria and other microbes, and also because of the very small genome sizes. Also, there is not a common gene across viruses, as in the case of the 16S gene for bacteria and archaea. Viruses are also made of double-stranded or single-stranded DNA or RNA, making it more difficult to study. Finally, sequence databases are usually biased towards either viral pathogens or very well-known bacteriophages or viruses that infect bacteria, most of which have previously been cultured. But even with all the difficulties of studying viruses, they are very important members of the human microbiome. They are present in anybody's site, including the gut, the skin, the bladder, the blood, and even the respiratory tract. It is estimated that viruses, particularly bacteriophages, outnumber their host by a factor of 10 to 1. Viral communities can respond and change in relation to health and disease, including, for example, periodontal disease and also inflammatory bowel disease, and also across various body sites. Viruses also change in response to antibiotic treatment, intimate contact, and diet, which can sometimes be associated and correlated with changes in their host relative abundances. Some viruses can also be acquired through the consumption of certain food items, for example, dairy products and plants. New studies are also shedding light on the ability of the virome to influence immune responses. We know that the virome is able to stimulate continuous immune responses without causing any symptoms. And this capacity has been documented for several different viruses, for example, the herpes virus and also the hepatitis B and C viruses. Some viruses can also be used for phage therapy and treat antimicrobial resistant infections. Although this is not really a new topic, it is now being revisited. Finally, the virome may provide answers to microbially mediated phenomena where bacterial studies have come up short. Another interesting fact about viruses is that some of them can be acquired through birth, although not really all of them. And it seems like moms continue to see the viruses throughout the baby's development. Also, as I mentioned very briefly, viruses respond to high-fat diets in mice. For example, it seems that viruses' lytic lysogenic cycles shift in response to a high-fat diet. More specifically, lysins, which are involved in lysine or killing bacteria, seem to be more represented in relation to a high-fat diet. This suggests that viruses may kill the bacteria that may be associated with a low-fat diet. As you may imagine, the implications of this in humans remain to be studied in more detail. Also, as I mentioned very briefly, viruses are present even in sites that were thought to be sterile previously. For instance, a study I had the opportunity to be involved in characterized a human urine virome in relation to urinary tract infections. Although we didn't see differences in terms of bacteriophages, it was probably one of the first studies to detect human papillomaviruses or HPVs in urine using high-throughput sequencing. And just to give you a brief background, none of the individuals had any abnormal pap smears or indications to have an HPV infection, but none of the HPVs detected in the urine samples were associated with low or high-risk infections, indicating that urine possesses HPVs that remain to be characterized and their roles to be determined. It is also very important to understand that there are over 150 different HPV strains, some of which we know can cause disease, but for most of them, we don't really understand their roles yet. Also, it is known that other sample types, such as cerebrospinal fluid, is a source of bacteriophages and other human and animal viruses, as you can see here in this particular study. Blood has also long been thought to be sterile unless there is an infection. Herpes viruses, aniloviruses, and pox viruses, all of them have been detected in blood samples. But this particular study from 2017 took samples from over 8,000 human subjects and characterized their viromes using high-throughput sequencing, probably one of the biggest studies characterizing human blood samples. The authors identified a number of human viruses, some of which were integrated. The authors also found evidence of contamination with reagents, which is a big issue when working with low biomass samples like blood. Viruses, particularly mammalian viruses, have been very useful in predicting disease risk. For example, this is a study characterizing the stool viromes of children at increased risk for type 1 diabetes. The authors first characterized the stool microbiomes of the children, showing that first, the vast majority of the viromes are composed of bacteriophages, as you can see here in green, and second, that enterovirus B or EBV was one of the viruses to be the most prevalent in the children's stool samples. The other very interesting discovery was that children with prolonged shedding of the same EBV serotype and children with consecutive positive results for different serotypes had similar higher odds of developing islet autoimmunity compared to children negative for EBV. As you may imagine, these findings have very important implications for disease prediction and disease treatment. Although not a recent topic, viruses can also be used to treat diseases that seem to be associated with bacteria, and this is also known as phage therapy. And this is a study published last year with a couple of very interesting findings. The first one was that the presence of acytolysine positive Enterococcus faecalis was correlated with the severity of liver disease and with mortality in patients with alcoholic hepatitis. The second very interesting discovery is that phages infecting these Enterococcus faecalis strains could actually reduce the disease severity. Again, as you may imagine, this opens a whole new venture into disease treatment and disease prediction. So as you saw, there is a lot of ongoing and future potential for viral metagenomics in areas where we want to, for example, understand the natural history of viruses, what is present, when the infection happened and where did it happen. If we want to continue to identify potential viral associations in unexplained illnesses. If we want to identify novel viral relationships with health and disease. If we want to identify potential novel emerging and re-emerging pathogens in veterinary medicine. If we want to identify viral associations as risk factors for disease, and very relevant for the ongoing SARS-CoV-2 pandemic, if we want to use viral metagenomics as a surveillance tool for zoonotic pathogens. With virome research, although very interesting and increasing, there are a number of biases that can be introduced into the pipeline, very similar to any microbiome study, and I'm going to be talking about some of these biases today. A virome pipeline usually involves several steps that are very similar to any microbiome pipeline, including sample collection, sample processing, sequencing, and bioinformatics. While there is not a good number of studies assessing this bias in virome research specifically, we can certainly learn lessons from microbiome studies. Let's talk about sample collection first. Sample collection and storage is one of the first steps after study design, and there are different practices for sample collection, and this will depend on the sample type. For example, the sample collection methods for saliva and urine might be different to those of stool samples because of the nature of the sample. But once a sample is collected, one major factor that can challenge microbiome studies is the storage temperature. It is widely suggested to immediately freeze a sample at -80 degrees for long-term preservation. But when a sample is stored at higher temperatures, for example, 4 degrees or even room temperature, there is the risk of variation in some of the results. Other studies may require a preservation buffer if there is no immediate access to a -80 freezer. Some of these preservation buffers include, for example, RNA later and omnigen gut. Also, sometimes freeze-thaw cycles can affect the abundance of certain viruses. So it's not really recommended to use a sample after multiple freeze-thaw cycles. Other suggestions may include making multiple aliquots after the sample has been well homogenized. There were a series of experiments performed at DNA Genotech testing the capability of omnigen gut to stabilize DNA viruses, specifically the crasphage and bacteriophage T5 in stool samples under different conditions. In parallel, they tested the detection of these phages with no stabilization at baseline, as well as room temperature at -80 degrees after 14 days. They also did very similar experiments with omnigen gut, where they tested the detection of these phages at baseline, at room temperature after 60 days, at 50 degrees after 3 days, and they also tested various freeze-thaw cycles. In terms of the baseline information, there were no differences in the qPCR results of crasphage and bacteriophage T5 when comparing fresh samples and the omnigen gut results. In terms of the crasphage, no differences were noted in any of the arms of the study. But in terms of bacteriophage T5, there was a significant decrease in CT values at room temperature after 14 days without any stabilization. These results indicate that while some phages may be persistent across different conditions, as in the case of the crasphage, other viruses like bacteriophage T5 may actually be more sensitive to changes in storing condition. As you may imagine, this may also translate to other viruses. It's going to be very important to select the best stabilization conditions depending on the type of study and using the appropriate reference materials like mock communities. Let's now talk about sample processing. Processing a sample for virome studies before sequencing is essential. A number of virome studies perform a filtration concentration step where samples are centrifuged first to remove any big debris, and it's usually followed by serial filtrations. The idea is to remove cells that are larger than the used pore size. Some labs also do a concentration step where viruses are actually concentrated using protein columns, and some other labs perform additional purification steps using, for example, cesium chloride gradient ultracentrifugation. Microbiome studies have shown the impact of host removal or the lack of host removal before nucleic acid extraction. In the case of bacteriophages, for example, it is essential to remove any bacterial cells and do a DNase and RNase treatment before nucleic acid extraction. And this is very important because there are phage genes that can be similar to those present in the bacterial host and may affect further data interpretation. Nucleic acid extraction methods also can affect virome results. There are a number of different methods and a number of different kits out there for viral nucleic acid extraction that each scientist will need to test. I'm going to be showing you a couple of examples of the effect of certain nucleic acid extraction kits using ATCC's virome communities. Some researchers also prefer to do an amplification of the nucleic acids before doing library prep using what is called random amplification. Studies using mock communities for internal use have shown that certain viruses can actually be overrepresented after random amplification. For example, single-stranded DNA viruses seem to be overrepresented when using what is known as multiple displacement amplification, or MDA. And finally, the method for library preparation, particularly those associated with capturing RNA viruses, can also add biases to the results. I'm going to be showing you some results about biases that can be introduced during this particular step in both previous studies and with some of our experiments using ATCC's virome communities. As I mentioned, the effect of various treatments before sequencing viruses has been determined. And this is a paper that is highly recommended if wanting to look at the effect of different treatment methods for viral metagenomics before sequencing. The authors first created a virome community composed of 7 DNA viruses, including bacteriophage lambda, here shown in black, the vaccinia WR virus, here shown in red, bacteriophage 529, here shown in light blue, the human adenovirus 5, here shown in purple, bacteriophage M13, here shown in light orange, the mice minute virus P, here shown in pink, and the porcine circovirus, here shown in green. And there were several different arms of the study. The first arm included no treatment. The second arm included 2 consecutive centrifugation rounds at 3000 Gs for 10 minutes. The third arm included a filtration step through 0.45 micromembranes. The fourth arm of the study included a filtration step through 0.22 micromembranes. The fifth arm included what is known as an iodixonal cushion, which is a method to concentrate and partially purify viruses. The sixth arm of the study involved a centrifugation step as described, a filtration through 0.45 micromembranes, and the iodixonal cushion step. The seventh arm included an amplification method known as CISPA, or sequence-independent single primer amplification, which was used as a full genome amplification method for unspecific detection of viruses. The eighth arm of the study included a method known as MDA, or multiple displacement amplification, which does not employ sequence-specific primers, but amplifies all the DNA at a constant temperature. Finally, the last arm of the study included a centrifugation step as described, a filtration through 0.45 micromembranes, an iodixonal cushion step, and the step called MDA. I know it is a very complex study, but a couple of things stand out from the results. First, when using a centrifugation step, the vaccinia virus, here shown in red, seems to be reduced, and similar results were noted when applying a filtration step through 0.45 micromembranes. One of the other things that stand out is the overrepresentation of the single-stranded DNA virus M13, here shown in orange, when using the multiple displacement amplification method, or MDA. When zooming into each virus in the mock community, there is, as expected, between 0.1 to 10-fold increase in the relative abundance when using either MDA or the CISPA methods. The take-home message is that there is no perfect method for the purification and isolation of viruses. Therefore, each scientist will have to evaluate the most suitable method for their study, preferentially using reference materials such as mock communities. In terms of the sequencing technology, although we have moved mostly to Illumina sequencing, there are still researchers that are using ion torrent sequencing. And differences in sequencing technology can add biases if trying to perform, for example, meta-analysis or combining data sets that are using different sequencing platforms. In addition, although we don't have the results of the effect of the sequencing depth for ATCC's virome communities, some of our results have shown that a sequencing depth of less than 3 gigs of data, which is about 6 million reads, can affect the detection of viruses, particularly at higher levels of classification. The effect of the sequencing platform has been evaluated for viruses, as I mentioned, and this is a proof article that should be published this year, where the effect of the Illumina versus the ion torrent sequencing platforms, as well as different sequencing kits, were evaluated. Starting from left to right, the sequencing platforms and sequencing kits tested included the Nextera XT, here shown in dark blue, the Nextera XT Nano, here shown in dark green, the Kappa Hyper Plus, here shown in light blue, the Kappa Hyper Plus Nano, here shown in light green, the Kappa DNA Ion Torrent 316, here shown in dark gray, the Kappa DNA Ion Torrent 318, here shown in light gray, the Kappa DNA Ion Torrent S5 510, here shown in orange, and the kappa DNA ion torrent, here shown in light orange. Although ideally a mock community would have been a more controlled sample, clinical specimens were used in the analysis, which were positive for various human viruses. As you can see here in this plot, the number of viral reads will depend on first, the type of virus, and second, the sequencing platform and the sequencing kit. For example, the number of enterovirus reads does not seem to be affected by the sequencing platform and the sequencing kit, as you can see here with the low variance, but the number of poliovirus reads, for example, seems to be affected by the sequencing platform and the sequencing kit, as you can see here with the larger variance. The virus coverage is also affected by the sequencing platform and the sequencing kit. As you can see here with this poliovirus specifically, the genome coverage seems to be the highest when using the Illumina MiSeq compared to the ion torrent sequencing platform. Finally, the bioinformatic analysis play a huge role in virus detection. One of the first things to do in viral research is to in silico remove any host sequence, including human, animal, plant, or bacterial sequences from your data set post-sequencing. This will ensure that you are analyzing viral sequences, and this will in turn significantly reduce the amount of computer power and time to process your results. Also, although I'm not going to be talking about assembly tools, assembly of virus reads can add an additional layer of biases to the results. The other main factor affecting viral bioinformatics, as you may imagine, is the annotation tool used. And I'm going to be showing you the results of 3 different annotation tools using ATCC's viral mock communities and the biases that can be introduced. One of the ways to address some of these biases and to benchmark reagents and bioinformatic tools is by implementing mock communities to the pipeline, either before or during the experiment. I would say preferentially, we would like to benchmark any reagents and any tools before the experiment, like we often do in a commercial facility. But reference materials like mock communities would need to continue to be included into a viral pipeline on a regular basis because there are still biases that can be introduced even when the reagents and the tools have been benchmarked. For example, factors such as changing the technician from day to day or issues during shipping of the samples to the lab, all of those things can affect viral results. And the only way to address these and other issues will be by including reference materials like mock communities. But the realization of including reference materials such as mock communities came up very recently. To the best of my knowledge, probably around 2012, 2013, maybe a little bit before that. And more researchers are acknowledging the importance of reference materials such as mock communities, but there is still room for improvement because, for instance, in articles published in 2 important microbiome and microbiology journals in 2018 alone, only 30% reported a negative control and only 10% reported the use of a positive control. So it's very important to implement and report the results of both negative and positive controls. The good news is that reference materials such as mock communities can be used as positive controls, but as I mentioned, mock communities are still not being implemented on a regular basis in microbiome studies, even though they are available through institutions, laboratories, and commercial facilities. As you may imagine, most of the mock communities available are intended for microbiome analysis targeting the bacterial fraction, and very few mock communities have been developed specifically for viral research. The good news is that ATCC has developed 2 different viral mock communities composed of either nucleic acids or whole viruses. And this is an application note where Diversigen had the opportunity to collaborate. I'm going to be showing you some of that data today. And just to give you a brief background, ATCC developed mock communities composed of 5 different human viruses with variations in their genomic composition and structure. For example, the mock communities possess double-stranded DNA viruses, including envelope viruses like the herpes virus, and also unenveloped viruses like the adenovirus. The mock communities also possess RNA viruses, including the positive sense single-stranded RNA virus like the Zika virus, a negative sense single-stranded RNA virus like the influenza B virus, and also a double-stranded RNA virus like the Rio virus. So as you can see, the mock communities have a very good representation of human viruses with great variation in genomic composition and structure. As I mentioned, we had the opportunity to beta test ATCC's viral mock communities, which are made of equal concentrations of either nucleic acids or the whole viruses. In the case of the nucleic acids, once we received the samples, the RNA was converted to cDNA, and the cDNA was pulled along with the DNA. And we had 2 arms for the library preparation process. One where the pool libraries would go through just an extra library prep, And the second arm included an additional step involving the amplification of the pool library using semi-random primers. Then we will go through sequencing and data analysis. And in terms of the data analysis, we used 3 different annotation tools, including MetaFlan2, Kraken2, and VirMap. In the case of the whole viruses, once we received the samples, the nucleic acids were extracted. In this case, we only tested 1 kit. But again, the RNA was converted to cDNA, and the cDNA was pulled along with the DNA. And we had 2 arms for the library preparation process. One where the pool libraries would go through just an extra library prep. And the second arm included an additional step involving the amplification of the pool library using semi-random primers. And again, we will go through sequencing and data analysis, and we used 3 different annotation tools, including MetaFlan2, Kraken2, and VirMap. Let's take a look at the MetaFlan2 results. MetaFlan2, just to give you an overview, is a marker-based analysis tool, and it might work better with very well-known viruses. Here on the left side of the plot, I'm showing you the expected proportions of the viruses in the mock immunities, followed by the results of each of the arms of the study. The second column shows the results of the viral nucleic acids going through just an extra library prep. The next column shows the results of the viral nucleic acids going through the additional amplification of the pool library using semi-random primers. The next column shows the results of the whole viruses being extracted with this particular kit. And the next column shows the results of the whole viruses being extracted with this kit, plus amplification of the pool library using semi-random primers. And several things stand out from the results. One of the first things is that the real virus was not detected in any other arms of the study. Second, the adenovirus seems to be overrepresented when using the amplification of the pool library using the semi-random primers, as shown here in blue in the third and last columns. Finally, on the fourth column, you can see a bias towards the herpes virus, here shown in green. In terms of the number of false positives, we identified a couple of false positives and a couple of false negatives, as we didn't detect the real virus. Let's take a look at the Kraken2 results. Kraken2, just to give you an overview, is a K-mer-based analysis tool. The second version, unlike the original Kraken, suggests that it has a greater sensitivity for virus detection. Again, here on the left side of the plot, I'm showing you the expected proportions of the viruses in the mock immunities, followed by the results of the different arms of the study. The second column shows the results of the viral nucleic acids going through the extra library prep. The next column shows the results of the viral nucleic acids going through the additional amplification of the pool library using the semi-random primers. The next column shows the results of the whole viruses being extracted with this particular kit. And the next column shows the results of the whole viruses being extracted with this kit, followed by the amplification of the pool library using semi-random primers. And several things stand out from the results. Again, the real virus was not detected in any other arms of the study. Second, the influenza B virus and the adenovirus seem to be overrepresented when using the amplification of the pool library using the semi-random primers, as shown here in yellow and blue in the third and last columns respectively. Finally, on the fourth column, you can see a bias towards herpes virus, here shown in green. In terms of the number of false positives, Kraken2 results seem to have the greatest number of false positives in all of the arms of the study. In terms of the number of false negatives, the real virus, again, was not detected in any other arms of the study. Finally, let's take a look at the VirMap results. VirMap, just to give you an overview, uses techniques that merge both nucleotide and protein information to taxonomically classify viral reconstructions independent of genome coverage and independent of read overlap. Here on the left side of the plot, I'm showing you again the expected proportions of the viruses in the mock community, followed by the results of the different arms of the study. The second column shows the results of the viral nucleic acids going through just an extra library prep. The next column shows the results of the viral nucleic acids going through the additional amplification of the pool library using the semi-random primers. The next column shows the results of the whole viruses being extracted with this particular kit, and the last column shows the results of the whole viruses being extracted with this kit, plus amplification of the pool library using semi-random primers. And several things stand out from the results. The first thing is that all of the expected viruses were identified using VirMap, including the real virus, and the proportions of the viruses in the nucleic acid mock community going through just an extra library prep had very similar proportions to those expected. Again, there are biases that are introduced during the use of the semi-random primers and during the nucleic acid extraction method with and without the use of the semi-random primers. In terms of the false positives, VirMap probably did the best job, only one false positive was identified in 2 out of the 4 arms of the study, and no false negatives were found as all the expected viruses were identified. These results show that viral communities are very helpful in identifying biases that are introduced during viral research. And here I just show you a couple of those biases. While we know now some of the applications of viral metagenomics and standards in addressing some of the biases that can be introduced at different stages of viral research from a microbiome and metagenomic point of view, I wanted to take the opportunity to talk about the use of viral reference materials for clinical applications that include the detection of ongoing and future human viruses. And I wanted to talk more specifically from our experience with the ongoing SARS-CoV-2 pandemic. As I mentioned, viral metagenomics can be used as a surveillance tool for the detection of SARS-CoV-2 sequences in various sample types. The ability to do so relies on the addition of SARS-CoV-2 sequences into the respective viral databases and, of course, of the annotation tool used. Here in Diversigen, we have the ability of detecting SARS-CoV-2 sequences in various sample types, although we have not done so with actual samples, rather publicly available metagenomic datasets. I mentioned very briefly the use of VirMap in some of our beta testing of ATCC's viral communities, and from the tools tested, it provided the best results in terms of the accuracy, including the detection of all the expected viruses and the very low number of false positives compared to other annotation tools such as Metaflan2 and Kraken2. And this is the reference of the VirMap paper. Once we added the SARS-CoV-2 reference genome into our database, we proceeded to identify and download available metagenomic datasets from NCBI testing positive and negative for SARS-CoV-2. As you can see here on the table, 5 samples were positive for the virus and 2 were negative. And these are bronchoalveolar lavage fluid samples, which are expected to be low biomass samples, or samples with very low diversity of microorganisms, including viruses. We then proceeded to profile the metagenomic datasets using VirMap. And as you can see, we were able to detect the genome sequence of the virus in all 5 samples testing positive for SARS-CoV-2, and we were not able to detect the genome sequences in the negative samples. This further illustrates the ability of the annotation tool used, in this case VirMap, in being highly specific. VirMap was also able to identify a number of DNA and RNA viruses in the samples, which is expected. As I mentioned very briefly when explaining the biases that can be introduced during viral research, the sequencing depth is one of the factors that can affect the detection of any virus, including SARS-CoV-2. To further investigate this, we evaluated the coverage and the recovery of SARS-CoV-2 sequences at 3 sequencing depths. When we look at a sequencing depth of about 1 gigs of data, we notice approximately a 3x coverage, as you can see here on the top part. In contrast, when looking at a sequencing depth of about 8 and 18 gigs of data, we notice coverages of around 12 and over 100x respectively. This data indicate that the deeper the sequencing depth, the more coverage you get as expected. But also this data indicate that each researcher will have to evaluate the specific sequencing depth needed depending on the aims of the study. Again, I strongly suggest the addition of reference materials such as mock communities in determining the best sequencing depth for your study. With any viral metagenomic analysis, utilizing this approach for the detection of SARS-CoV-2 in metagenomic datasets needs to be accompanied by standards and reference materials. ATCC, for instance, has a number of SARS-CoV-2 reference materials available that will be very, very helpful. And I would like to add that in the case of studying SARS-CoV-2 using viral metagenomics, it will also be very helpful to add other reference materials like ATCC's viral communities to further understand how the mentioned biases apply to SARS-CoV-2 research. The application of SARS-CoV-2 and other reference materials can help determine the biases that can be introduced when, for example, testing a nucleic acid extraction method, or when testing reagents that are used for the conversion of the viral RNA to cDNA. Also, I know a number of researchers and colleagues that are developing qPCR assays for the detection of the virus in various sample types. For example, a number of qPCR assays are being developed for both clinical and environmental samples, like sewage samples, as part of the surveillance efforts to detect the virus. In this case, SARS-CoV-2 and other reference materials can be used to validate a qPCR assay. These reference materials can also be used to validate a metagenomic assay, starting from sample collection down to data analysis, as I mentioned. They can also be used to develop novel surveillance assays for the detection of the virus. And last but not least, SARS-CoV-2 and other reference materials can be used to study the evolution of the virus if the reference materials represent the original isolate. And these are just some examples of the use of reference materials in SARS-CoV-2 research and other viruses. So again, I strongly suggest that each researcher uses these and other reference materials to the best of their knowledge as needed. So how do we apply these standards and reference materials moving forward? We know that viruses will continue to affect humans in one way or another in the future. So we can continue to use these materials to expand our viral databases, which will help in surveillance efforts. With viral reference materials, we can continue to validate current methods and current reagents for collection, stabilization, extraction, and detection assays. With these reference materials, we can validate and benchmark novel reagents and novel approaches for sample collection, detection, among many others. We can, for example, layer information into metagenomic datasets to provide new biological and clinical insights related to viruses, including SARS-CoV-2. But moving forward, I envision that a number of currently available reference materials like the VirMap communities will continue to evolve as the scientific community believes is the most appropriate depending on the type of study. Next iterations of mock communities may include other viruses like bacteriophages, for example. It is also possible that next iterations of viral mock communities will be more simple or maybe more complex in order to mimic different sample types and different scenarios. And with this, I'm very happy to take any questions.
Well, thank you, Tasha. That was a really insightful talk. In just a few moments, we'll begin our Q&A session. Please use the chat function available through the webinar program to submit your questions. The recorded webinar presentation will be available on demand on the ATCC website at www.atcc.org. Okay, so let's start with our first question. This one looks to be around sample processing. What are the best practices for nucleic acid extraction leading to viral metagenomics?
That's a very good question. Ideally, in a more conservative virome lab, filtration concentration steps are applied before extracting the nucleic acids to ensure that you have captured as many viruses as possible. Ideally, you should also remove any host cells and any free nucleic acids using a DNase and RNase treatment before extracting the nucleic acids. Other labs would do additional purification steps like cesium chloride gradient ultracentrifugation, but I think moving forward, the best practice is to add reference materials like mock communities.
Okay, good. Now, as a follow-up to that question, what typical viral fractions can be expected?
That's a good question. So it's going to depend on the type of filtration, concentration, and purification steps if they were applied. If you also amplify the nucleic acids using random amplification, for example, that can increase your viral fraction. But if you don't apply any of those steps, and if we're talking about relative abundances without any of these steps, you can expect less than 1% of the reads or the sequences to be viral.
Okay. Now, this next question, this is an interesting question. Do we know to what extent is the virome personal? So the asker goes on to say, for instance, bacterial strain carriage appears to be relatively stable and tends to be fairly personal signal. So what degree might be the same for viruses?
So that's a great question. There was a paper from one of my previous institutions at UC San Diego where they characterized the oral virome, and the title of the paper is literally "Human Oral Viruses Are Personal, Persistent, and Gender Consistent." So they basically found that viruses are very specific, and if you plot the data in a picoa plot, the viromes tend to cluster according to subjects, and the viruses can even be traced up to 60 days in individual people. And there was another study that found that the majority of oral viruses are also found in very few individuals. And those viruses that have single incidences are the most widespread. So I would imagine that this would apply to other viruses like gut viruses and skin viruses.
Okay. Now, our next asker wants to know, how do we move beyond pathogen bias in our reference databases? And as follow-up to that, what is needed to ensure that we capture the most deadly, common, and/or costly viruses?
That's a good question again. I think there's going to be a greater interest towards those viruses that can cause disease for various multiple reasons. I think what we need to do is to take microbiome data and microbiome results and move towards a more translational stage. And a great example of this is the discovery of the CRAS phage back in 2013, 2014 by using bioinformatics. And one of the things that stood out from this particular virus, which is not a deadly virus, was that it's present in a huge number of subjects. And then we saw a number of studies utilizing the sequence data to predict the potential bacterial host, because this is a bacteriophage. And it turns out that scientists were able to point to the bacterial host to be a Bacteroides species. And this enabled the isolation and the characterization of the virus. So I think moving forward, we can take examples like this one to keep building our viral databases.
Okay, great. Now, this next question, Tasha, is going to require you to look into your crystal ball. What are your predictions for the future of viral metagenomics, discovery, and surveillance given the current pandemic?
That's a great question. I think the fields of viral metagenomics, discovery, and surveillance will expand now that our attention is mostly towards just one virus. We know that by focusing on SARS-CoV-2, for instance, we can use viral metagenomics as a surveillance tool if we have the appropriate reference genomes in the respective databases. Like I mentioned, other viruses will continue to affect us in one way or another in the future, so it's going to be essential to apply the lessons learned these days for viral metagenomics discovery and surveillance.
Okay, good answer. For our next question, this sounds to be kind of a technical question. What kind of sequencing depth is needed to achieve good profiles in different sample types, such as human stool, blood, or environmental samples like wastewaters?
So this is really going to depend if you have done any of the steps like filtration and concentration before extracting the nucleic acids. In an academic lab, that is usually the practice, but I haven't seen any studies addressing the sequencing depth needed to capture the diversity of viruses in different sample types after applying all these steps. But in a commercial facility like Diversigen, however, these steps are usually not applied because they are not high throughput. Although here in Diversigen, we at least use methods to capture both RNA and DNA viruses. The sequencing depth needed to capture viruses will depend on the diversity and the complexity of the sample. For example, a stool sample is going to be more complex than a biopsy sample. And the sequencing depth will also depend on the extent of host contamination if you haven't applied any host removal step. You would imagine that a biopsy sample, for instance, will have a greater degree of host contamination compared to a stool sample. So in this case, although a high diversity is not expected in a biopsy, it has a huge extent of host contamination. And looking at our results here in Diversigen, we have seen no changes in viral diversity at sequencing depths greater than 5 gigs of data in both stool samples and in some cases biopsy samples.
Okay. Now, can a tool like VirMap be used to find new viruses in sequencing data?
That's a good question. So the short answer is not really. We have seen with internal data that you can identify viruses up to 96, 97% similar to those in the databases. But any virus with an identity percent lower than that will not be captured by VirMap. So for virus discovery, we will need to probably use approaches that are similar to those done for the discovery of the CRAS phage back in 2013, where the data was assembled and further validated and characterized. But there are also tools out there for virus discovery, for example, Vibrant, that do not really require similarity to viruses in current databases.
Okay, we've got some more questions coming in. Here's one. What are the best ways for genomic library preparation to cover both RNA and DNA viruses?
That's a very good question. I think as of now, I don't know of any studies addressing the specific library preparation protocols for both RNA and DNA viruses, but I think that would be a great study moving forward.
Okay, Here's a good one. How do you resolve the rapid evolution and diversity of viral genomes for data analysis?
That's a great question. I think the best approach would be to keep on building our databases and use tools that would enable not only the identification of viruses, but also the discovery of viruses, like, for example, Vibrant.
Okay. Now, when using a mock community as a reference, is it your experience that the more viruses, the better, or can you have too many viruses in a mock community reference standard?
That's a great question. I think we are at the beginning stages of using viral reference materials in viral metagenomics, so there's not really a correct answer. A good representation of viruses with differences in structure and genomic composition is a great start to assess biases and standardize a virome pipeline. And of course, like I mentioned, moving forward, these reference materials they will be continuing to be evolving to include, for example, other specific viruses.
Okay, good. Now, why do you believe there is such drastic results when using different library prep methods?
Yeah, that's a good question. Again, there is not a good number of studies addressing different library prep methods for viruses, but I believe the differences that we saw, at least here in Diversigen, when beta testing ATCC viral mock communities, were due to the original purpose of the semi-random primers, which were targeting specific viruses within the viromes.
Great. Now, it looks like we've got time for one more question, and this one is also around future directions. Where do you see the next important clinical applications as it relates to virome research?
I think the possibilities are endless. The next clinical applications, I think, lie on developing new detection and new surveillance assays for not only viruses that we have right now, but also for potential future viruses.
Okay, great. Well, at this time, we will conclude our Q&A session. I'd like to thank Tasha for the excellent presentation, and thank you everyone for attending this webinar. One final word. Although there's already a wealth of information on the human microbiome, much is still unknown. As scientists begin to move towards the next wave of methods, research will be challenged with managing burgeoning data sets and parsing information into meaningful insights. Optimization at each step of the process via authenticated standards is essential to the validity and reproducibility of your data. That's why ATCC is committed to providing the authenticated microbiome standards and data analysis solutions needed to produce reliable and reproducible data. ATCC microbiome standards provide a solution to this problem. From sample collection to data analysis, ATCC microbiome standards enable you to optimize your diverse research applications with confidence and improve the consistency and reproducibility of your data run after run. For more information about ATCC microbiome standards, navigate to www.atcc.org/microbiome. Thank you again, and have a great day.